diverse audience, and I expected something along those lines, so I will try to keep my talk high level, but at the same time give you a flavor of what types of approaches we are using within the field of network science, and also what they can be used for. Question one, I promised to ask you some questions, right? Question one, what do social systems such as Facebook and Twitter, or technological systems such as the internet, or infrastructure that enables cell phones to communicate, or information systems such as the World Wide Web, or transportation systems such as the airline routes, FYI, this is direct. When there you see kind of black line, that means that there's direct flight from Atlanta elsewhere, and focuses on Atlanta as the major hub in the US. Or biological systems such as brain, or the cell, and even financial systems such as stock market. What do all of these seemingly different systems have in common? Yes? The keyword was in the title. Hint. I don't expect necessarily that you will know the answer. Can you guess? Yes?
They're all like some form of network.
They're all some form of network, meaning they can be modeled with these, what we call nodes. Think of it visually, but think of them visually, but it's not visually that we're inspecting anything. You have these nodes such as A, B, C, and D, and you have these links or interactions or edges between the nodes that are modeling some kind of interaction or relationship between the objects. So I'll give you illustration of what networks corresponding to some of these systems are. In social networks, nodes are people, and Facebook communication or real-world friendships are interactions. In brain networks, nodes will be different brain regions, and if two brain regions fire up, meaning are active while a person is performing the given task, that means the regions are correlated, so you link them. In transportation networks, you can have anything and everything, such as if there is a direct flight from one location to another, you put a link between the two locations. And in biological networks, such as the protein-protein interaction networks, nodes are proteins, and if they physically interact, and meaning bind in the cell, you link them with an edge. So this talk is about networks. But before I go into that, I want to tell you a little bit about who I am and how I got to be where I am today, because, well, I was kind of suggested to include a little bit of career advising into the talk, and just so you understand, we're all people. I'll tell you a little bit more about why I believe networks are fun and important, and hopefully I convince you that's the case. I'll talk to you about general research features that my group is working on across multiple domains to illustrate to you all the different applications network science can be used in, and I'll go to at least one, maybe two, some more specific research projects depending on the time. Everybody has a story, so here's mine. I'm from Bosnia-Herzegovina, this country colored in orange on the map of Europe that used to be part of ex-Yugoslavia about 20 plus years ago. I'm from the capital of the city called Sarajevo. This is the famous Pigeon Square. I really don't know how to translate the original name from Bosnian language into English. It's a very popular old town destination from, we were under Ottoman Empire for 6 centuries, a long time ago, of course. So you can see lots of cultural influences still in the old town. In '84, you were not born then, or most of you were not born then, we hosted Winter Olympic Games. I'll go back a couple of slides. So you see the mountains surrounding the city. The city is kind of in a valley, surrounded by mountains, so we qualified for this. However, early 90s, for 4 years, my hometown went through, well, actually the whole country went through a horrible war. I was in the center of the war for a full year before we decided it's time to go. 3 more years the war lasted. Because of the war, which happened when I was in 4th grade, I had to move, and then the war expanded, and I had to move again. And then during this period, I was a refugee, with 7 of us living in a one-bedroom apartment for 3 full years. So if you think things are bad, things can get worse. But still, if you ask me whether I had a happy childhood, I can only remember the good things. So I guess that's there's still lots of scars from the war, but I remember mostly the good things. And just when I started, or was about to start high school, we went back home. So I actually finished the whole high school in my hometown. Now, college time came, and I was like debating what direction to choose. I love math. Computer science barely existed at the time. I mean, it did, but not on large scale, so I didn't really know what it was. And I liked biochemistry, drug design, whatever, bioapplications, and I really had a tough time choosing, and one of my high school teachers basically said, computer science is about to explode. Trust me, choose that. And I was a straight A student most of my life, so I would be good at anything, even arts and letters. But I really kind of was debating between these three fields. So I chose computer science. And my degree was given within the School of Electrical Engineering back home, so it was a little bit strange organization. There's no unique formula where which department is located, as I'm sure many of us know. And if you are wondering how old computers, basically how undeveloped the field was at the time, I got my first computer when I was in second year of college. And this was type of screen monitor that I had. I don't know if anybody has ever seen this student-wise. Now, I finished my undergrad degree, and I knew last year of college, I told my brother, I want to go and do a PhD and become a professor. And I chose to do it in the US. Why? Well, in my home country, really, I think you can still feel consequences of the war in terms of economy. Not necessarily as many perspectives to advance as I would like. I could have chosen developed parts of the Europe. Don't get me wrong, my country is developed, going there for vacation is amazing, but depending on the economy, not so much. But in Western Europe, you are mostly expected to pay for your own PhD degree, whereas in the US, you are given first, everything's paid. And on top of that, you're given a scholarship, order of $30,000 a year, which can cover all of your expenses. So all they require you is to work hard. Did you know that if you're doing PhD, at least in computer science field, not law, not medical degree or anything alike, in computer science, PhD degrees are paid. So maybe something to consider. Now, during my undergrad, I had 4 semesters of Cisco Networking Academy, connecting routers and connecting and configuring routers and switches to enable the computers to work. So I was like, of course I want to do that. I know how to do that. I want to do my PhD in the area I'm familiar with. So it's called computer networking. And even though there's part network in the word, it's very different than what I'm doing right now in terms of network science. And then my future advisor at the time emailed me from UC Irvine. Her name is Natasha Krajur. Happens to be from ex-Yugoslavia as well. Coincidence? I don't think so, but still. And she was like, you want to do bioinformatics? You have background that I'm looking for. You want to do bioinformatics. My reaction was, I don't want to clone people. That's as much as I knew about bioinformatics. So, I typed email saying, thank you so much. I read a couple of your papers. I don't really understand the bio field. And I almost clicked send. And then my cousin, who was doing a PhD at Caltech at the time, basically said, if you decide to go to that field, you'll get into the PhD program. It's very hard to get into a PhD program, especially as an international student. So I took a risk. It was the best decision of my life. Bottom line, don't be afraid of something you're not familiar with. Trust in yourself that you can do it, and you will be able to do it. So I started my PhD. I didn't formally go to master's degree. I gained it along the way of getting my PhD. I started in 2006, and I got my PhD in 2010. Most of the, on average, PhD in the US lasts between 5 and 6 years. I did mine in 3.5 years, actually. And people were like, well, either you didn't do enough or something's off. I can tell you for the first 2 years, I worked 15 hours a day, 7 days a week, because my student visa depended on me being successful. And I really didn't want to go back home, if you want to think of it that way. So my 3.5 years actually translate into normal people's 5 to 6 years, so don't think of it as anything special. But remember that last year of my college, I told my brother I want to go to PhD. I said I would finish it in 4 years, knowing how long it would take, because I had a cousin who was going through the process. And I will try to get a professor position in the US. In Europe, it doesn't matter. And that's why I got my PhD from UC Irvine. And then my brother reminded me in 2010, when I became the Notre Dame faculty, I'll come to that in a moment, that I actually said that. So I really achieved my 5-year plan at the time, and I'm very, very proud of it. So in 2010, I became assistant professor, and this year I was promoted to associate professor with tenure. If you don't know what tenure is, it's the biggest milestone in the faculty career, if you're on tenure track position. So they asked me during the interview, do you like football? And I was like, no, but if you don't want to hire me because of that, I don't want to work for you. And they gave me an offer. So I have 3 jobs. I do research, I teach, and I do service. And imagine my research is in intersection between computer science, math, and biology. Remember the 3 fields I was debating between when I was going for college? Well, I ended up doing all 3, which I absolutely love. These are kind of names of my research fields or fields. I'm not going to go through them. I could even add data mining, I think, in there. Teaching-wise, I teach a required sophomore course for our computer science and computer engineering majors. It's a math course, it's required. They take it. It's the first computer science class they take. Not a favorite class, not a favorite course. Network science is open to both seniors, undergrad seniors, as well as grad students, and I think it's a lot more favored than a math course. Service-wise, I service numerous committees, and I also do a number of educational activities with the goal of increasing participation of minorities, both gender-based minorities, racial minorities, in computer science. As a woman, I would like to see more women in the field, but be aware that it's not just women's responsibility to attract other women to computer science. Men need to do the same. The same holds for racial minorities. And if you're wondering why we need diversity, because diversity makes everybody more productive. Okay? This is my group. Well, this is a picture. Actually, most of these students are gone by now. To date, I've supervised a number of undergraduate students. I am proud of my undergraduate student supervision record. Of all of them, almost everybody went to grad programs at schools such as MIT and Stanford and Carnegie Mellon. And Jen just graduated this year. Yes, I lost track. There's a number of them, and look where she is. The same place where Terry Nesco graduated 2 years before, and I'll actually interview Jen, speaking of small world. This is Bo. Bo and Willie are my 2 most recent undergraduates that have graduated, and Willie went to MIT to their famous media lab, and Bo went to Stanford, and actually Bo informed me this week, or last week, about a week ago, she was meeting with Melinda Gates, so pretty cool opportunity. These are were my PhD students that graduated late, Fazl and Yuri. Vipin and Joseph are still in my group, and on top of that, I have 4 other PhD students, newer ones, and I have a couple of undergrads as well. Most of the work I will be talking about today is based on work by students who are already done. My research is funded. I want to acknowledge these funding sources before the end of the talk in case I run out of time. NSF, Air Force, NIH, and Google. Is pizza here? Not yet. Okay. This part will take about 5 minutes. So if pizza arrives, you will have to suffer for at most 5 minutes. So going back to networks. We already covered. They are everywhere. Any system in real life you can you can model with networks. Who cares? What? So they're everywhere. Who cares? What am I showing here? Question number 2. Don't say pencil and ring, please. What am I showing here? Yes, please.
The carbon atoms make graphite as well as the diamond.
Perfect. Graphite and diamond, and both composed of exact same elements. So it's not carbon atoms that make graphite soft and dark and diamond clear and hard. It's how they're linked. Networks matter. Imagine this being a network of a pathogen that you take a medication, within a week you're good. You're not sick anymore. And this is a pathogen from the same species, just different strain. It's drug resistant and it kills you. Well, we need to they have the same genes. So it's not the genes or proteins that are, well, it's not the genes that are different. It's how they produce proteins and how those proteins interact with each other that's different. This is what used to be X years ago, 6 years ago or something, my Facebook friends wheel. Each of the nodes on the edge of this circle is my Facebook friend. Or what used to, I actually did recently, removal of some people, I can't recognize, kind of. Anyway, if 2 of my Facebook friends are also friends, then there's a link between them. Question 3. What can you tell about me just by looking at this picture? Yes, I will tell you the answer, and you can be sure that is it, but think of my background. I told you about my origins and everything for a reason, so what would this mean? If you didn't know anything about me, could you hypothesize what's going on? What's the interesting trend? Let's answer that first, and then we'll answer why it happened. Do you see anything interesting? By interesting, not like fun, just something weird, unusual. Yes, please.
There's like an entire arc that has no connections.
Reword it, kind of take the complement of what we just said. If there's this part that doesn't have connections, there are 2 separate parts that do have lots of connections. So we have a top where many people, many of my friends know each other, and bottom where many of my friends know each other. Now, what could those 2 groups of people correspond to? Yes, please.
You're friends from Europe and you're friends from America.
Perfect. I lived in 2 completely different geographic locations. So that could be a hypothesis. That's exactly what's going on here. You will see, you will see a node right here. And this person's connections to all of these people at the bottom are clear, and similar happens on top. That's a friend of mine that started PhD from Bosnia, from my home country, that started PhD in my advisor's group a year after I started. So she shares lots of common friends with both of my friends' groups. And even if you didn't know this, you could have guessed something on those lines. I had to guess that it was a spouse or a significant other at the time and close. That would be a valid hypothesis. Okay, let's do a survey. I want everybody to raise your hand at one of 2 answers. Answers being left or right. If these nodes are people and edges are friendship, would you be in the left network? Raise your hand if so. Look around. A couple of hands. Or would you be in the right network? Raise your hands. Look around. But I didn't finish. There's a deadly virus going through. Would you be in the network on the left? I'm seeing some changed minds. Or on the network on the right. I'm still seeing somebody who likes fast spread of disease. Okay, I changed almost everybody, though not everybody's mind, by giving you just additional information. Bottom line is, whether more is more or less is more, meaning the number of connections, patterns matter depending on the context. Now, yeah, we studied, I gave you a couple of examples of how networks are useful, but in real life networks are so complex, they look like this, and know that this is a small network, it has only 1,000 nodes. Think of nodes, networks with millions of nodes. They look like these hairballs. You cannot do anything by eyes, observe any interesting trends by eyes. So what the best we can do is develop efficient methods for analyzing networks, computational, mathematical models, formulas, whatever we are trying to measure. Then we need to efficiently implement it, code it so that it runs fast enough, and then we can recognize interesting patterns in the networks. Is the pizza arrived? Okay, I will not remind everybody that pizza's not here. If the pizza arrives when I get to my next outline item, just scream at me, yell at me.
We've got at least 15 minutes.
Okay, so I think at that time I will be probably around here, or we'll see. Okay, so now I want to, rather than, when we have an idea what networks are and how complex they can be, I want to give you an idea of what it is that my research group works on. We work on theoretical projects. Given a network, what is a pattern? What is an interesting signal in the data, such as a triangle or a square or something like you see here? We call these graphlets, by the way. It's one of the methodologies we are working on that can efficiently link triangles to disease and squares to something that promotes health. I'm just giving you kind of dummy illustrations. Bottom line is we use these structures as a mathematical model for quantifying the structure of a network and then linking that structure to the given functional behavior. By that I mean phenotype such as sick and healthy, or graphite and diamond, or whatever else you can think of. We developed new methods for what is called network clustering. Given a network, there are groups of nodes that are somehow linked together. Linked, not necessarily linked directly like it is here. You see clearly blue nodes are densely interconnected with each other. Densely means lots of possible edges exist. So are purple, so are whatever these colors are. But in between the groups, there's very few edges. So these would be clusters of nodes, groups of nodes that are densely interconnected. There are other notions, mathematical notions of what it means for nodes to be similar that would make them belong to the same cluster. But the hypothesis is then that this group of nodes are co-workers, this group of nodes are friends, and so forth. There's some characteristic that these nodes share that they don't share with any other group. And that holds for every group defined. I'll touch on this topic a little bit more in detail, but the bottom line is when you have two networks, Facebook and Gmail, you don't know which Facebook has which Gmail address, or Gmail and text, Gmail and cell phone numbers. You don't know which Gmail address is associated with which cell phone. Well, now we do because of advances in technology. In biology, this could be network of one species and network of another, and you really don't know what gene in one species corresponds to what gene in another species. There are hypotheses, but it's directly related to the notion of alignment. So when we don't know what nodes in one network correspond to what other nodes in the other network, the goal is to find the mapping so that you conserve lots of network structure. And I will try to illustrate that using my hands. If the center of my palm is a node, and all of the ends of the tips of my fingers are nodes, mapping, and these are two networks, this would be a good mapping. This would be a bad mapping. You want to conserve network structure. Why? Well, because if you know a lot about this network, and you don't know anything about this network, meaning I will illustrate you applications of in disease, aging, or whatever, we cannot study some species, we cannot study some systems, meaning Facebook, if it releases the data, it will strip up all information about the nodes. Maybe you want to learn something about nodes. Then by finding these similar network regions, and knowing a lot about this region but nothing about the other, you just transfer the knowledge between them because they share structural similarity. Data in real life is noisy because of because of disadvantages and drawbacks of technologies that are used to collect the data. So, given a network, like toy network like here, the goal of link prediction is find edges that are reported but shouldn't be there, and find edges that are missing, and predict them that they should be there if that's the case. And the goal is, we are making some predictions from these networks. So if I have noisy data, I'll make noisy prediction. If I can clean the data, I will clean the predictions. Data is highly heterogeneous. Here is the illustration of gene layer of the cell. Genes produce proteins which are then actively acting in the cytoplasm of the cell. Metabolism, proteins act as enzymes in metabolic reactions. So all of these slices, and the same holds for social systems, all of these slices, all of these data types are different slices, functional slices of the same system, namely the cell. So the goal is how do we efficiently integrate the data so that when we make predictions from the integrated data, we are more accurate than doing so from individual data types. Networks are dynamic. So this is the same network that evolves over time. Edges form, edges break. In social science, in social networks, just think, I just removed some of my Facebook friends breaking those links. So the goal is, given that we know what happened up to this point in the history of a network, I can mine some patterns, and based on them, can I predict who will interact tomorrow or a month from now or a year from now? Can we predict the future? We are really asking that question. I'll at least cover this. We'll see how much time we have, and maybe I'll go a little bit into details into dynamic network analysis. And even those I'll try to keep high level, but just to dig deep to give you a little bit more. On applied side, I hope we all remember high school biology and the central dogma of molecular biology. The DNA is in the cell, in the nucleus of the cell. It gets transcribed into messenger RNA, that it gets translated into a protein. Protein is what is actually active genetic code. It's DNA genes are passive genetic code because they're in a nucleus. So after a gene, the DNA expresses a protein, synthesizes a protein. Protein is what goes and does things in the cell. My apologies if this contradicts detailed biology, because I've been argued against before if I'm not being precise, so I'm keeping this very high level. And then once proteins are done functioning, they get degraded, so that there's no over-concentration of a protein in the cell. So I work on the notion of synthesis of a protein, so on this end, and that is done by machinery called the ribosome. And this is collaborative effort with Patricia Clark from Notre Dame, where we now not model the whole cell, just the given protein. This is an illustration of a 3D structure of a protein. We model this 3D structure as a network. There's a way to do that. And by studying network patterns as opposed to raw structures or raw sequences, remember that DNA is a sequence of 4 nucleotides, protein is a sequence of, well, up to 4 nucleotides and up to 4, here up to 20 amino acids. These are just sequences of letters that you can manipulate. But proteins do fall into 3D structure, which we can then nicely model as a network. On the other hand, I also work on protein degradation by the machinery called the proteasome, and this is collaborative effort with one from the University of California, Irvine, who I also worked with when I was a PhD student, together with my advisor. But most of my work is when proteins are actually active, and what happens for various phenotypes such as aging, disease, such as cancer, how can we improve therapeutics, make drugs more efficient, personalized medicine. Will we be able to carry our genetic data on our cell phones? Soon we will. It's gotten so cheap to get your bio data, and then we can direct, target, not we, biomedical scientists with the help of our applicants, let's make that clear, to make treatments personalized per person. There's no magic formula that works for everybody. That's why there's side effects for certain medications. Because it works for somebody, but not for everybody. And I will try to give you an idea of what we do in the context of aging. And it directly links to the 2 theoretical projects. Now, what I also work on are many other domains. I'll give you 2 examples. Social networks. We've given to our Notre Dame students, undergraduate population, free cell phones with free data plan for years. And we've actually recently expanded this to Fitbits, so we can collect other types of data. So, we gave them all of this for free. In exchange, free. Nothing's free. In exchange for collecting their SMS, phone call, email, Facebook communications, and the proximity as measured by Bluetooth, which is a proxy for face-to-face interactions. We don't collect any content. We only collect which type of interaction happened at which time, and we have years of data on this. Now we are measuring physiological signals such as heart rates, sleeping patterns, and so forth. If you think something's free, oh, absolutely, it's not free. But this has all been approved by all appropriate protocol committees and whatever, so there is no ethical constraints. So what we are trying to answer are many different things, but I'll give you one of the most popular things we still don't have, nobody has an answer to this, but people are trying to approach this. Do we interact because we are similar? Or do we become similar because we interact? It's a chicken and egg problem. And probably both happen. But how do you disentangle them and figure out when what happens? And I can't imagine all possible implications of being able to predict what's going to happen, who's going to interact with whom, what. Both physical, so when I say similar, means both physical appearance and personality traits. We have a number of traits on each of these people. So very interesting questions we can ask, but that's kind of the key motivation. Now, why is Fitbit important? I'll tell you why. It measures physiological signals that you cannot objectively necessarily report. So I work on what we call physiological networks. We are the first study, to my knowledge, that model physiological data networks. And I'll tell you the key idea and the key outcome, skipping all the methodology in the process. data was shared with my group by Sidney DeMello, actually from my department as well. 18 subjects were given 89 images, shown 89 images. And their physiological signals, namely galvanic skin response, which to my understanding it's very correlated with the amount of sweat that you produce or something along those lines. They were shown happy images, scared images, angry images, different types of images. And they were asked to report how they feel. Are they happy when happy image is shown to them? Are they scared when a scary image is shown to them? And we have some ground truth knowledge on how they should feel based on the experts in the field of psychology, not computer science. And we have shown through our framework that we can better tell you how you feel than you can by objectively analyzing your data, then you can tell us subjectively. Now, if you wonder about applications of this, we have not gone, and I'm over-exaggerating just a tiny little bit, but big follow-up would be to prove that this actually holds. We have done first step in the direction. But long-term applications are, and I'm not saying we're going to pursue this, my goal is to develop methods for applied scientists to then use that to pursue similar ideas. Imagine if you want to detect somebody's behavior if they're trying to hide it, such as terrorists at the airport or something along those lines. Huge number of applications that research of this type can have. So, the only thing I have is to dig into at least one, if not two, research projects. We'll see how I am after the end of the first one. I'll begin by motivation, what behind the computational approaches we are developing. Who wants to live longer, healthier lives? Emphasis being number one on healthier and only then longer. Perfect. Everybody. Now, if I'm a biomedical scientist and trying to study an individual from the moment they're born till the moment they die, who's going to die first? I know it's a little bit of black humor, but if everything goes well, I will, because I'm already an adult. So, because of long lifespan and a number of ethical constraints, we cannot study human aging experimentally. Of currently 263, it's around 260, genes in human that have been implicated in aging. Only 3 have been confirmed experimentally, by that I mean wet lab, by medical scientists, to be involved in aging. The rest are what I think of computational and statistical predictions in the following form. Remember, genome, genes are sequences of letters. So I can study aging experiment, not me, by medical scientists, of course. Aging in model species such as mouse, even worm, even fly, even yeast. Our cells look the same. There's a membrane, there's nucleus, there's cytoplasm, there's lots of evolution that's being conserved. So if I know that this gene in model species is involved in aging, all I need to find is a gene in human that has similar sequence of letters that match well. Hypothesis is that these sequence matches correspond to evolutionary conservation, and then you can transfer the knowledge about aging to this particular gene. Most of the current knowledge comes from these types of, from what is called genomic sequencing. A data type that you can relatively easily measure, by easily I mean both efficiently in terms of both resources and time. Resources mean money and time. Is gene expression data. Basically at different ages of one's, now these are not coming from the same individuals. Typically they're coming from different individuals because of the length of human types. This is done on humans. Your gene expression data basically measures the activity of a gene or a rate at which a gene produces the given protein. The rate at which a given gene is active and actually functioning in the cell. So if a gene is active up to some point and then becomes inactive, or if it's inactive and then becomes active, as long as there's some change in activity patterns through gene expression analysis for a given gene, it's implicated as aging related. Because it is aging related, something changes about the gene with respect to gene expression. However, remember, connections matter. Genes, that is proteins, don't function in the cell on their own. They interact in concert with other proteins to make us alive. So, to address the biggest issue with both of these types of approaches, this was my first grant I proposed, was to draw analogs of genomic sequence alignment through network alignment. This is not a new idea, just using network alignment in the context of aging was the new idea of the grant. Basically, given we have these networks of different species, so instead of finding sequence-similar regions, find network-similar regions based on network alignment. I already explained the intuition behind that. And then we follow the same thing. Transfer knowledge between the aligned network regions. Or, let's pretend for a moment we have networks for human at different ages. We don't, I'll tell you how we address that. But let's say that we did. Then I just study evolution of network structure with age. And I will tell you what that means in a little bit more detail. And if a gene, if the gene's connectivity patterns significantly change with age, the gene becomes more and more connected with other genes, or less and less connected with other genes, then we implicate that gene is aging related. Importantly, network alignment uses sequence alignment as part of the problem. So we inherit the best of this world plus more. And how we get to these networks, the problem for any species, most species of human, is there are lots of drawbacks in biotechnologies for detecting interactions. It's very easy to detect dynamic gene expression data. For dynamic network data, we don't even have the full static network. We have a reasonably complete static network. So what we can do is combine the static network with this dynamic gene expression data to only keep at a given age part of the big static network that's active with respect to gene expression. So again, you use this data in the process of dynamic network analysis. So you get the best of this plus one. And I'll start by discussing this first, and I will present some mathematical challenges. Why this is a hard problem and why it's important to develop cool and efficient methods to solve it. Given two networks, big networks, the goal is to, again, align them, find the mapping between their nodes that conserves lots of structure. This is the figure you've seen, just more zoomed in. Again, fine, we don't know that T corresponds to E prime, A corresponds to A prime. Our goal is to map these nodes, for example, like shown in the figure, mapping is shown in the dashed lines. And then the goal is to try. This is a completely conserved region. This is partially conserved region. This is a conserved edge, but this edge is not conserved because it does not exist between E prime and A prime. Nonetheless, we find approximately similar regions and then transfer knowledge from this network to another. Network alignment has a number of applications, and I'll list just some from studying human aging or any other biological process that you can easily study in model species. And model means species that are easy to manipulate in the lab. Human is certainly not one of those. Between any model species and human. Social networks, mapping. There's a cool study, in the interest of time, I'm going to skip it, feel free to talk to me about it later. This is just an illustration of how they've used network alignment to actually show you can never properly de-anonymize the data. So let's say that Facebook tries to give for research purposes real data, part of their Facebook network, and they want to make it, of course, fair to you, so they will not reveal anything about you. Well, it turns out, just by giving us that network, I can contrast it to the publicly available Facebook data with all the known labels, check your Facebook profiles, make sure all the info is private, and I can reveal who you are, and more importantly, based on if your information is public, but your friend's information is private, based on all their public friends, meaning public accessible to the public, I can actually infer something about them. So you having your information public is not fair to your friends who have their information private. Because based on you, I can learn about them. Keep that in mind. Computer vision, matching objects. Again, application is if you have a video and a face, can you match to determine where a given person is? Person of interest, did anybody watch the show? It's a spooky, spooky show that reminds me of my research every day. I can't do anything along those lines, but theoretically it's possible. Ontology matching, link prediction, and so forth. Here's the mathematical problem. Network alignment has subgraph isomorphism as a special problem, subproblem. And don't worry about subgraph bar, just think of a graph bar. Graph isomorphism is nothing but given two graphs of exact same size, same number of nodes, same number of edges, can you map their nodes so you conserve all edges? So, in other words, this is a square. Ignoring A, B, C, D, and ignoring 1, 2, 3, is this a square? Think about it. I'll give you 10 seconds. I saw your head immediately nodding. Can you just tell me why it is a square?
You can rotate the bottom two nodes on the right.
So think of flipping 3 and 4, you get a square. Or in other words, I map A to 1, B to 2, C to 4, and D to 3, and I get the same network. So I conserve, so these two graphs are isomorphic. There is a mapping, this one, that conserves all the edges. Are these two networks isomorphic? And give me an answer only if you're willing to bet $1 million on it. I actually had some people lose the money. Of course, I am kidding, but really I want you to give me an answer only if you're super certain. And students have been wrong. People have been wrong before, not just students. I think it's been 20 seconds. Who has an answer? Because they're confident about it. Okay, I can tell you they're not, but that's not the point of what I'm trying to illustrate. 4 nodes, seconds. 10 nodes, well, it's 25% bigger, so it should take me only 25% more time. I can give you 5 minutes without pen and paper and looking through every possible mapping and determining that no such mapping exists that conserves all edges. That's the only way to prove these are not isomorphic. Well, there are other ways. But bottom line is, I give you this, it will take you trillions of years. And these are networks with approximately 1,000 nodes. I give computers networks with millions of nodes. It will take I take all the computers in the world, put them into one gigantic supercomputer. And I ask them for two, not this large, very large networks. Are they isomorphic? And I'll actually refer to subgraph isomorphism now. It doesn't matter, very similar problem. It will take computers trillions of years to answer the question. Can we wait that long? Obviously not. So what do we need to do? We need to come up with approximate methods that will still be very accurate within certain bounds of accuracy, but reasonably fast. In biodomain, we can definitely wait for hours. In online advertising, once you open the web page and an ad shows up, they have to give you a correct ad before you enter the button. Milliseconds are important. So in certain domains, such as bio, accuracy is important. We can wait days. In other domains, they're willing to accept more errors at the expense of speed. So it's domain specific. Bottom line is, but it doesn't matter which of these two scenarios you're dealing with or anything in between, we can't wait trillions of years. So that's what we are doing. Now, I can't tell you how we are doing it for one method. It will take me probably half an hour. But I will point you to a couple of papers if you're interested. Browse through them. This is one of our prominent recent methods called Magna. And then we implemented it in user-friendly, hopefully, user-friendly software where the user only needs to embed the two networks and the name of the output file. This is for advanced users who are actually actively involved in the field. And just to illustrate to you the accuracy of our approach, which is shown in red compared to state-of-the-art approaches, and note that this was paper from 2014, so we have newer methods, but this is still just for illustration purposes. The height of the bar is accuracy. These are results for synthetic and real networks. Synthetic meaning we know the answer. We always test first, and when we know the answer, how accurate we are. Here we don't know the answer, so we have approximate measures of how we do it. The higher the bar, the better the method, and we beat all the methods we tested against it. They were state-of-the-art at the time. And on this topic, we have a bunch of other methods and papers, and if you're interested in this topic, this is the paper I will start with, because it's among the recent ones, and it's a survey of not my work only, but all the work in the field up to 2015. And since dozens and dozens of new methods have appeared, so it's a very popular field. Now I'm going to stop because pizza is here, and I can even end actually, because I am close to time, so let me briefly jump through this. I'm not going to talk about the problem of how we construct the networks. Please just understand this is the only thing we do have in terms of network, static network. I want this is the slide that is the most relevant. But we combine it with age-specific gene expression data. Which genes are active at which age? So we get age-specific networks, where each network contains only the genes that are active according to gene expression data and their interactions. And then we study the evolution of network structure with time, with age, globally, overall, Are things different? No. 3 different tests say all of these networks globally are similar, which means good, our cell doesn't fall apart. But when you zoom into certain genes very locally rather than the whole network, we found 5% of all genes, about 500 of them. They either gain connectivity, meaning gain connectivity, or they lose connectivity. And we hypothesize them to be related to aging. I'm going to skip next couple of slides and tell you just about the validation of these. We found our predictions. You go and check, do they perform aging-related functions? And gene ontology and disease ontology are just databases, which proteins have which functions and which proteins are involved in which diseases. So yes, we perform our predictions are enriched, meaning many of them, significantly many of them perform this function, which is directly what we wanted. And these are the 4 diseases that we find enriched in them. And in articles, this means PubMed ID, database for bio-focused research papers. In these corresponding papers, we linked each of these, not we, the authors, linked each of these diseases to aging directly. And then we go and take the most confident of our predictions. That doesn't exist anywhere in any database. How do we validate them? They don't exist in databases as having certain function. Well, you go and dig into literature, because maybe there's a new article that implicitly or explicitly linked to aging, and we managed to validate 80% of the genes we tested in the literature. I'm going to skip next couple of slides and just remind you, ask me any questions about career, being a faculty, why academia over industry, why industry over academia. Networks are cool, you can do lots of stuff with them, including studying aging through different types of computational strategies. I'll stop here. I already acknowledged everybody involved in the work. Thank you for your attention.